Please listen carefully. కొంచెం కన్ఫ్యూషన్ గా ఉంటుంది. Gastrulation. Gastrulation, it is a dynamic process, Nana. Gastrulation is a dynamic process. So already in the last class we discussed about bilaminar embryo. Bilaminar embryo completed. Next, here bilaminar embryo means here two layers or cells, formative cells or embryonic stem cells are arranged in two layers. Of course, this upper layer, don't forget this trophoblast or tropoectoderm. It will be there always. Okay, trophoblast. Listen carefully, trophoblast. And here this upper layer of embryonic stem cells, this is epiblast. And this lower layer, it is called hypoblast. Okay, so this is bilaminar embryo. Now it is undergoing some changes in these layers. Okay, so this epiblast continuously it is keeping on dividing. Okay, now so cells divides and comes downwards, and these cells pushes this hypoblast like this. See, your cells which descends from this epiblast occupies the space of hypoblast. Now this is called endoderm. So this layer which is pushed down by endoderm, this is called hypoblast. Okay, now further this hypoblast develops into yolk sac. Okay, it develops into yolk sac. Please remember yolk sac. Okay, other hand, it is also called as extra embryonic mesoderm. Right? Now here cells again divides and they descends like this. These cells are called mesodermal cells. Okay, we will discuss later about mesodermal cells, but let us see here this embryonic disc. This is lateral view, beta. This is lateral view. If we see the upper view here on the middle longitudinal axis of this embryonic disc or blastodisc, here primitive streak will appear. Okay, primitive streak. Here this is lateral view. Now I am going to show you upper view. Here this is viscoblastula. Okay, here two layers are there. Epiblast that side, hypoblast, and even yolk sac also there that side. Please remember that side. Okay, so this is middle portion of this disc, and longitudinally a streak appears. Okay, here important point is there. Appearance of this primitive streak, it is the beginning of gastrulation. Okay, so appearance of this primitive streak, it indicates the beginning of gastrulation. Important point, appearance of this primitive streak, It indicates beginning of gastrulation. Okay, so this is called primitive streak. Primitive streak. Primitive streak. Next, along with this primitive streak, a primitive groove. A groove appears. Okay, cells always keeping on dividing. So here a groove-like structure, longitudinal groove appears. This groove is called primitive groove. Primitive groove. Okay, see better. Primitive groove. Just imagine primitive groove is just a canal-like structure. Primitive groove, and at this anterior part, a shallow structure appears. This shallow structure is called primitive pit. Primitive pit. Okay. At the anterior part of this primitive pit, the swollen part, this is called Henson's knob or primitive node. Henson's node, I think. Henson's node. Primitive knob. KNO. Primitive knob. Okay, like different names are there. Primitive, just a second. Primitive knot, primitive node, Henson's node, primitive knot, or primitive node, like different names are there. Okay. Next, along with this primitive groove, here lateral fold-like structures are there. These are called lateral folds or primitive folds. These are primitive folds. So such kind of changes taken place in the embryo during gastrulation. Here formation of the primitive streak, it is the initial stage, or it indicates the beginning of the gastrulation. Gastrulation, it is a dynamic process. Dynamic process means changeable process. So here cells undergo some changes. So here like this structure will appear. Okay, this is called gastrulation. Okay, gastrulation. Okay, if we see all the here further changes, formation of trilaminar embryo, formation of notochord, formation of nerve cord. Okay, all changes will take place here. This place only, this place. Okay, here it is upper view, and if we see the lateral view here, this place looks like this. Epiblast. This is hypoblast. Sorry, endoderm. Endoderm, and here this another layer which develops into yolk sac. This is called hypoblast. Okay, of course this is trophoblast. Trophoblast. Okay, beta. So this is lateral view. Okay, this is lateral view. This is upper view. Upper view. Okay, your primitive streak appears on the epiblast layer only like this. Primitive streak. Okay. So this process is called gastrulation. Gastrulation. Come on, let us see next formation of trilaminar embryo. Okay, see before this here separation of these layers, it is called delamination. Delamination, separation of what is that, endoderm from the epiblast, and this pushes down this hypoblast. Entire this process it is called delamination. Okay, right, please remember. Next, let us see formation of trilaminar embryo, where all these changes are taking place in this region. Okay, so whatever we are discussing here about the further changes of embryo, here they are taking place this region only. Please keep it in your mind. Okay, right? Yeah, okay now. Next, formation of trilaminar embryo.
Sir,
Yes.
here all epiblast cells will migrate and form endoderm, or
Yes,
few
yes, yes. Only epiblast cells migrates and forms endoderm. Okay, now such kind of migration of all cells, it is called morphogenic movements. The movements of the epiblast cells towards downwards. Okay, now it is called morphogenic movements. Morphogenic movements of cells. Next, here this entire process of formation of gastrula. Okay, this is gastrulation. Gastrulation. Okay, now next, say this is epiblast, and this is endoderm, epiblast, endoderm. Okay, and here this is trophoblast. No, no, don't forget this layer, trophoblast. Trophoblast. Next, already this layer will be there. Hypoblast. Hypoblast. Next, here again and again these cells undergoes division. So here some cells comes down and forms here mesoderm. Mesoderm. Before this, here some carda mesodermal cells of epiblast. Please remember, carda mesodermal cells. Carda mesodermal cells. Carda mesodermal cells of epiblast descends down and they forms a mass of cells. This is called notochord. 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 Okay. Next, here some cells, epidermal epiblast cells, some cells forms neural plate. Neural plate. It becomes into neural plate. See, observe carefully. Neural plate. Some cells modifies to form neural plate like this, and these are common cells, epiblastic cells. Here this is neural plate. Next, at this stage, here these cells divides into number of cells, and here appears a neural groove. Okay, see these cells invaginates downwards, okay, and forms neural groove. This is called neural groove. This neural groove develops like this. These are epiblast cells. Okay, are you following or not? Finally, it becomes like this. This is epiblast. It separates from the epiblast. This is epiblast cells. Epiblast. Okay, these are neural cells. Okay, finally, it becomes nerve cord. This is nerve cord. Nerve cord. Okay, beta. So here, like this, nerve cord will form. This is the formation of nerve cord. Various stages of nerve cord formation, right? 1, 2, 3, and 4. So finally, a tubular nerve cord will form. Tubular nerve cord. This is tubular nerve cord. Okay. Next, here cells from this epiblast descends like this. Here these cells are called mesodermal cells. Finally, this layer mesoderm will form. Mesoderm. This is called mesoderm. Now, this epiblast is called as ectoderm. Okay? Is it clear? Here, formation of neural cord, nerve cord, formation of notochord. Okay? Okay, so this process it is called neurulation. Formation of nerve cord, notochord, it is called neurulation. Neurulation. Once again, please listen carefully. Here, corda mesodermal cells of epiblast. Corda mesodermal cells of epiblast invaginates and becomes a mass of cells and forms notochord. Notochord. Okay. Next, some cells of epiblast undergoes modification and becomes neural cells and forms neural plate. Invagination of these neural cells induces the formation of neural groove. Neural groove gradually deepens and here finally forms a tubular nerve cord. Tubular nerve cord. So this process is called neurulation. Neurulation, right? Okay. Next, here cells descends down and here they forms mesoderm. Here this is endoderm. Okay, na? These are mesodermal cells. Now, after formation of this mesoderm, this epiblast, now it is considered as ectoderm. See beta here, 3 layers are there. Ectoderm, mesoderm, and endoderm. Now this embryo is called trilaminar embryo. Trilaminar embryo. 3 primary germ layers are formed. Ectoderm and mesoderm and endoderm. Okay. Next, here tubular nerve cord also formed. Notochord also formed. So all these changes will take place where Henson's node. Okay, now at the part of Henson's node, right? Please observe once all these stages. We will go to further step. Right. This is yolk sac. Hypoblast develops into yolk sac. This is separate diagram. Right?
Sir,
1 minute.
sir, నేను అనురాధ మేడంని.
Differentiation of mesoderm and formation of coelom. Say this is ectoderm and this is endoderm. Please observe carefully. And this is yolk sac. Yolk sac. Here this is tubular notochord. This is notochord. Okay. See, mesodermal cells here they descends like this. They extends between ectoderm and endoderm. Okay. These are mesodermal cells. Okay, mesoderm. Next, between this ectoderm and endoderm, here these cells are mesodermal cells divides into 3 blocks. 3 blocks. See beta, here this is a block, this is another block, and this is okay. 3 blocks. 1, 2, 3. So the mesodermal cells which are distributed around notochord and notochord. These are called epimere. This block is called epimere. Mesodermal cells which are present around notochord and notochord. Here this is called epimere. Next, this last one, which is near to the endoderm, this is called hypomere. Hypomere. Between these 2 layers, 2 blocks, this is called mesomere. Mesodermal cells forms into blocks. Okay, epimere, mesomere, hypomere, which are around the notochord and notochord. This mesodermal cells group is called epimere. The mesodermal cells which are present near to the endoderm are called hypomere. Next, mesodermal cells which are present between epimere and myomere, they are called mesomere. The block is called mesomere. Okay, again this epimere divided into 3 blocks. Epimere again divided into 3 blocks. Just this is hypothetical diagram data. Okay, epimere again divides into 3 blocks. Here block 1, scleral. These blocks also called as somites. These blocks also called as somites. So this somite, first somite, it is called scleratome. Scleratome, and this is called myotome or dermatome. And this is called myotome. sclerotome, dermatome, myotome. Epimere again divided into three blocks: sclerotome, dermatome, myotome. Right? Next, say sclerotome in further developmental stages, it develops into vertebral column. Vertebral column. Okay. See here, nerve cord and notochord like structures are there, no? So that region, it is divided, it is called as sclerotome block. It develops into vertebral column. Dermatome, it develops into dermis of skin. Dermis of skin and other connective tissue. And other connective tissue. Please remember, don't forget this. Dermatome develops into dermis of skin and other connective tissues. Next, myotome. This myotome develops into voluntary muscles. Myotome develops into voluntary muscles. Right? So this is about epimere. Epimere again divided into three blocks: sclerotome, dermatome, myotome. Sclerotome develops into vertebral column and dermatome further develops into dermis of the skin and other connective tissue. CT means connective tissue. Connective tissue. Right? Next, myotome develops into voluntary muscles. Next, mesomere and hypomere. See beta, mesomere. This part, mesomere, in further developmental stages, develops into urinogenital organs. Urinogenital organs means kidneys, urinary bladder, ureters, and reproductive organs. Urinogenital, excretory organs and reproductive organs, and their ducts. These parts derive from mesomere, mesomere of mesoderm, right? Next, hypomere. Last hypomere is there. Hypomere. See beta here. Hypomere. Just I am drawing here. This is hypomere layer. Hypomere. Two layers. For example, just let us think. Two layers are there. Here, this is endoderm. Endoderm. Of course, further developmental stages, endoderm develops into alimentary canal or digestive system. Next, this is called hypomere. Hypomere. This is hypomere. This hypomere divides into two layers. This is inner layer. What I am drawing here. This is outer layer. This is inner layer, outer layer. Okay. This is planktonic layer, inner layer. This is outer layer. Okay. See. Outer. What is that? Parietal peritoneum. Somatic peritoneum. Somatic layer or peritoneum, and the inner splanchnic layer. Between these two layers, coelom. Between these two layers, coelom will form. Body cavity. Okay. So in this way here, all our body parts derive from these blocks at early embryonic stage. Okay, this coelom, this is coelom. Coelom is extended between somatic layer and splanchnic layer, which derives from hypomere block. Hypomere block. Here, this is coelom. Between these two layers, this is coelom. Okay. So commonly these two layers are also called as peritoneal layers. Peritoneal layers. Okay. Somatic layer or parietal peritoneum, splanchnic layer or visceral peritoneum. Already we discussed in our first year classes. Somatic layer, it is also called parietal peritoneum. Splanchnic layer, it is also called as visceral peritoneum. Between these two layers, always we can expect cavity or body cavity or coelom. Okay, so in this coelom, particularly in higher vertebrate, this of course in invertebrates also, coelom is restricted around pericardium. Pericardium means around the pericardium, around the heart, and pleura, pleural, around the lungs, next to perirenal, around the kidneys, like that, and around the brain, like here the coelom, this cavity restricted around some important visceral organs. Okay, beta. So this is called differentiation of membranes, mesoderm, differentiation of mesoderm, and the formation of body cavity. This topic is differentiation of mesoderm. See beta, here mesodermal cells are differentiated into three blocks: epimere, mesomere, hypomere. Epimere, mesodermal cells which are located around nerve cord and notochord. These are called, this block is called epimere. See, mesodermal cells which are present around nerve cord and notochord. It is called epimere. Okay. Next, again, epimere divided into three somites. Three somites. These are called somites. These blocks are called somites. So these somites, first one, sclerotome, myotome, sclerotome, myotome, dermatome. Sclerotome develops into vertebral column in further developmental stages. Dermatome develops into dermis of the skin, which is also called as largest organ of our body, and other connective tissues also derives from dermatome. Myotome, here voluntary muscles of our body derives from myotome. Next, another block of mesodermal cells, mesomere. Okay, from this mesomere, urinogenital organs and their ducts will derive. Mesodermal block, mesomere, gives rise into urinogenital organs and their ducts. Right? Next, last layer, which is these mesodermal layers, which is present near to the endoderm. Here, they divides into two layers: outer somatic layer, inner splanchnic layer. Splanchnic layer inner means it attaches with the endoderm. Okay. And between these two layers, your body cavity will present. So this body cavity restricted to heart and lungs and kidneys and brain like important organs, right? So this is differentiation of mesodermal cells and formation of coelom. Differentiation of mesodermal cells and formation of coelom. Right, Anna? Okay? Carry on. Copy this diagram.